I tell people when I get sick, I want to be treated, but I'd rather just not get sick. And I think it's going to be easier to slow aging than it is to treat most of these diseases of aging.
So hello and welcome to the Cheeky Science Show, where in this video I had the absolute pleasure to interview Professor Brian Kennedy, who is currently working at the National University of Singapore at the Centre for Healthy Aging, where his lab is using multiple animal models combined with human studies to understand the mechanisms driving biological aging and to develop interventions designed to extend health span and lifespan. Prior to this position, Brian was the president and CEO of the Buck Institute for Research on Aging in California. In this interview, we discuss Brian's interests, current work, and excitement for future research avenues, so I hope you enjoy it. But first, it's time to deanimate. Great. So hi, Brian. Thank you very much for taking the time to talk with me today, and so welcome to the Cheeky Science Show. So I believe that you're joining me today from Singapore, and so we'll talk about the work that you're doing there later on in this interview and some of the exciting clinical trials that you're conducting. But you've also got a very impressive track history of research that you've done through your career so far. And so I guess my first question is, what got you into the field of aging? Where did that initial interest come from? And to you, what is the aging process? How would you define aging?
Yeah, I think, well, thanks for inviting me. First of all, it's a pleasure to be here, and we're right in the middle of a thunderstorm, so if you hear loud noises, that's why. I think there's two answers to why I got into aging. One was that I was an only child, and I grew up in sort of with all of my older relatives lived forever. I mean, both of my grandmothers reached, one of them was 99 when she died, and the other one 101. And so I got to watch a lot of people go through the aging process that were close to me. And people were at different states of health. The one grandmother was very healthy. She lived alone all the way to 101. And the other one had everything wrong and different attitudes. And so I was just curious about what causes aging. I mean, everybody says, oh, it's just natural. But to me, that doesn't make it good, right? So, I was curious about it. And then when I got to graduate school, another graduate student and I, Nick Austriaco, joined Lenny Guarenti's lab at MIT. And we wanted to work on yeast cells, but we wanted to pick the craziest thing we could think of to do with yeast cells. And at the time, there were two choices. One was aging and one was apoptosis. And apoptosis seemed too crazy because we're talking about a single-celled organism undergoing programmed cell death. And so we chose aging, and that started the whole process. The funny part of that, though, is that the other graduate student became a priest, and then he became a priest biologist and has a little lab, and he studies apoptosis in yeast. So there's a whole field on that, too. I guess we would have been okay either way. If you want how I think about aging, I think that it's really funny because the field is made a lot of progress in some areas. We've come up with numerous interventions that might slow aging. More recently, we've identified, and by we, I mean the whole field, has identified biomarkers of the aging process that are extremely useful. I think that like 30 years ago when I started in the field, everybody thought, well, it'll be relatively easy to understand mechanistically what causes aging, but to do interventions will be really, really difficult. And now we've got all these interventions, and I still don't think we really understand what mechanistically is causing aging. So it's a bit of a surprise. And the way I think about it is that, aging is not just one thing. It's not DNA damage. It's not misfolded proteins. It's a combination of things. And they can be read out in different hallmarks or pillars of aging. Inflammation, adult stem cell function, metabolism, macromolecular damage, all of those things. But what's really happening to me is that as you get older, you have a homeostatic network in your body that keeps you healthy. And it responds to the damage that's happening to you. And so, yeah, if it's somebody who's 40, we can measure aging. They don't run as fast. They probably don't think quite as fast if we do careful assays. But they're still highly functional. They're still active. They're still, very much on top of everything they do in their lives. What happens though is when enough damage happens, that network breaks down and it can't compensate anymore. Then you kind of spiral out of control. And depending on the risk factors you have and your lifestyle, you start getting diseases. So maybe you'll get diabetes, maybe you'll get cancer, or maybe you'll get some sort of cognitive impairment or some combination thereof. But to me, the interventions that slow aging are really maintaining that network. And so then it becomes a systems biology question. Why does intervening in the TOR pathway preserve the network? And I don't think we have a really good answer for that yet. It's one of those challenges because the complexity of that question is maybe beyond the ability to reduce it to a simple series of statements. And so the AI approaches may be very meaningful in that regard. I think that, but I would just come back to say that I don't think there's one cause of aging. I think there are lots of things that drive aging. Some things are more important in one set of individuals than another. And the interventions seem to improve all outputs of the network. They reduce inflammation, they improve proteostasis, but what their immediate effect is that's causing aging to slow down, I think is still an open question.
Really good answer. And I completely agree. I mean, one of the things that fascinates me most about the aging process as a kind of biochemist is just how complicated it is, but also how everything seems to be interconnected. And it just seems such an interesting process to study. And so you spoke about interventions, and some exciting research that was published last year was on alpha ketoglutarate, which is an endogenous metabolite that was shown in this, I think it was a cell metabolism paper, to extend both healthspan and lifespan in mice. And so could you tell me a bit about what alpha ketoglutarate is and what that research study found?
Yeah, so this is a central metabolite. If you've had biochemistry, you looked at alpha ketoglutarate, it's right in the TCA cycle. And actually, it's at the interface of protein catabolism and carbohydrate metabolism. So it's really helps with metabolic flexibility. It helps the cell convert. If there's too many proteins and not enough carbohydrates or the vice versa, it helps to convert among those different pathways. It can do a lot of things at the cellular level. AKG is a substrate for almost 1,000 different reactions. In some ways, it's a lot like NAD, that it's involved in central metabolism. It's involved in many different reactions. The levels of AKG go down with aging, and supplementing them back up seems to have a beneficial effect. And what we found is that if we start the mice at 18 months of age, it only extends lifespan by about 10%. But perhaps more importantly, it reduces frailty by 50%. So what we think is happening is compressing morbidity. It's shortening the time when you're sick. And if that phenotype is preserved to humans, that's a really exciting molecule, because that's what you really want, right? As long as health span as possible. And we're hopeful that AKG might help contribute to that effect. And there's clinical studies ongoing right now to try to address that.
Ooh, and so the other thing you noticed in that study was that there seemed to be sex-specific differences. And so I was wondering if maybe understanding why there were sex-specific differences could help to understand the mechanism of action of AKG in the aging interventions.
I think that it's really interesting that all of the interventions seem to have sex-specific effects. So AKG works a little bit better in females, but for frailty, it works in both. Rapamycin and metformin seem to work better in females. 17-alpha estradiol, for instance, works better in males. And we don't really know why. There's been some studies now, and it seems to involve sex hormones, as one might expect. But I think there's a lot of work to be done. I assume that some of those differences will be preserved in human intervention studies as well. So it wouldn't be surprising as we start testing interventions that some work better in males or females. And I think you're right. That's a very interesting clue to try to understand mechanistically what's going on. And it also points to the role of sex hormones in the aging process.
Exactly. And so the other thing that in that study, you used the calcium salt version of AKG. And so I think when the study came out, I did some calculations to work out the percentage of calcium by mass of Calcium-AKG. And so as well as giving the mice AKG, they're also getting quite a lot of calcium. And so I was wondering, to what extent do you think calcium might be influencing the results, or have you looked into that?
We have now, and it seems like you can get the effects with other forms of AKG. But the one challenge that I would bring up is that in the mice, we're putting AKG in the food, and they're eating all day long, and at higher quantities. In the human studies, we're using a sustained release version of the pill. And I think that's important because we want to get it more of it through the gut and create more bioavailability. So especially if they're taking, less on a per mass basis. So our evidence suggests that the time release version of AKG may be more beneficial in the human context, and that's something that we're testing. I don't think we have any support for the idea that calcium itself is generating the effects.
Okay, and so you mentioned the human clinical trials, and so I believe that there is in the works, or at least in process, a human clinical trial at Singapore looking at the effect of AKG on a variety of different aging biomarkers. And so could you tell me a bit more about the clinical trial, where you're currently at, and what different markers are you trying to look at?
Yeah, there are two clinical studies that are in process. One is using the Rejuvant product by the company, and so that's Calcium-AKG sustained release plus a vitamin. For men it's vitamin A, and for women it's vitamin D. That's what the product has, and that's a 6-month clinical study, people 45 to 65 years old. It's nearly finished, and I think it'll be finished in the next 3 months.
Cool.
And we'll be able to start to really look at the data. We're looking at methylation clock, as well as a number of clinical markers, inflammatory markers, metabolic markers, etc. And we're really hopeful that we'll see some beneficial effects on biologic aging. Now, we already know that that study is being conducted at Indiana University, actually. We already know that people that buy the product, they can buy a methylation test at the beginning, and they do a test 4 to 6 months later. And we are seeing a reversal of biologic age in those individuals, but that's not a clinically controlled study. I mean, it's subject to placebo effects and everything. And so it's great that we're seeing that, but we want to see how much of that translates through to a controlled clinical study. And then in Singapore, what we're doing is a whole series. Right now we're doing cross-sectional measures of biologic aging. So we have 300 individuals, and we're measuring probably 10 different things that address biologic age. And we want to begin to understand which of those work in Singapore ethnicities, but also to see how they relate to each other. So if the methylation clock says you're old, we have another clock from 3D facial reconstruction. Does that say you're old as well? Because I don't want to spend $5,000 to measure aging. I want to spend $200, but I'm not sure any one clock is sufficient either. So we need to know where the overlap is, where the uniqueness is in these clocks. And then then we're going to be doing a whole series of interventions, one of which is the sustained release calcium alpha ketoglutarate without the vitamins, because here I want to get more toward mechanism and not have the complicating factor of having the vitamins. But we'll also test a number of other supplements and lifestyle interventions. And the goal is really to figure out which interventions work the best in humans, because I don't think anybody really knows the answer to that yet. And also to figure out which ones work in which people. Maybe some interventions work better in males or females, or maybe some things don't work in smokers, etc. We want to start to personalize these interventions so that we can have a better efficacy in the long run.
That sounds really exciting. So when these results come out, what would you regard as being, positive results? And given if there are positive results, what would you do with that information in terms of what do you hope to do in terms of subsequent trials or investigations? Yeah,
I think that, there have been a couple studies already. There was a recent study on exercise and lifestyle interventions that showed that using a methylation clock, they could reverse aging by 3 years, I think, with a short intervention. And there was a previous study looking at taking 3 different small molecules where 9 people did it, and they could reverse biologic age by a couple of years. Those are really small studies. They're not controlled very well. However, I think they're important because they're suggesting that it might be possible to reverse aging, at least to some degree. I'm not going to say that David Beckham's going to be on the soccer field again, but but it may be that, I think a lot of people thought you could slow the process. But if you slow the process, even with the biomarkers, it's a really hard thing to measure because, over 6 months, they don't change that much. If you can actually reverse them, even by, 2, 3, 5 years, that's a really exciting finding. And that's what these studies are suggesting. And what we need to do now is go back and do really controlled studies to validate that. And maybe it is possible to reverse aging to some degree. I think when I started in the field, a lot of people said you'll never modify aging, it's too complex. And if you do, you'll just slow it down, you'll never reverse it. And now I think most people are saying it's possible to slow aging, but I don't know if you'll be able to reverse it. So, we're making progress. I don't know where the endpoint is, but even if we can extend health span by 5 years in the population, that's a huge economic and quality of life benefit. So I think we can set that goal first. And even if we just get there, we've already done a lot. We're going to have to do, these studies are relatively small, 120 people or so, 6-month interventions, people 45 to 65. If they show really positive data, then one of the things we'll have to do is to do bigger studies to validate and to get more personalized data. But, a lot of what we're testing is supplements and lifestyle. So we're really looking to partner with Singapore government and other agencies here to scale things into community testing. I would really love for everybody when they hit a certain age to get their biologic age measured. And if something as simple as a 3D facial reconstruction works, which is totally non-invasive, you could imagine that being done when you go to get your ID renewed, right? So I'm hopeful that we can really implement some of these things and start to have an effect. And Singapore is an ideal testing ground for that. Huge problem with aging here because it's a small island. There's not the reproductive rate is extremely low, and people live a long time. So it's almost like Japan in terms of the percentage of people over the age of 65. A little bit behind, but catching up. And so this is a great place to see if we can really do something about it.
Cool. Then that's really exciting to hear. So one thing that I'm interested in, as well from the kind of biochemical perspective, is potentially combining different interventions together. And so I know in one of the earlier AKG studies, they did it in worms, and they found that AKG alone extended their lifespan, but if the worms were dietary restricted, they didn't see as much of a benefit. And so I wondered what your thoughts are on not necessarily just AKG, but actually combining different interventions together and whether or not they could be synergistic or potentially detrimental.
I think I'm positive about this approach in the long run, but right now, I don't think we can predict what's going to happen. We've tested a large number of combinations in mice, and we've never found anything that's toxic, but we do find things that cancel each other out. And some things don't seem to interact at all. Other things are additive, and I honestly can't predict what's going to happen when we start the experiment. So, there are a lot of hackers out there that are taking 15 different things, and there are pills now that are coming out that give you 12 different things to slow aging. I honestly don't know what happens when you take 12 different things. And I think that's where we want to get to, maybe not 12, but we want to get to multiple interventions to maximize the effect. But I don't think we really know how to do that yet. And the other thing I'll say is if you get a pill that has 12 things in it and it's this big, ask yourself how much of each one could it possibly have? Because the AKG pills with the vitamin A are this big, and we'd like to make them smaller, but we have to make them that big to get the product in. And now, if you imagine 12 things, so I think that the anti-aging, market is still full of a lot of things that probably don't work. Having said that, there are some things that are very intriguing: the AKG, the NAD precursors, and some of the other stuff. And I think that it's fine for people to go ahead and start trying things. I encourage them to educate themselves and try things. But, we have to really validate this and figure out what's working in the long run.
Exactly. No, I completely agree. That was a really good answer. And so the other thing that, besides you're right about whether or not combinations would work or not, and as well talking about different doses of the interventions, the other thing that I always find quite interesting to think about is the time of day in which an intervention might be underdone, because the circadian rhythm like regulates so many different processes within our bodies. And so I was just wondering, have you looked into whether or not like AKG may be more beneficial if it was done in the morning or the evening or anything like that?
Yeah, again, in the mice, it's in the food, so they're getting a steady dose of it. We'd have to change the dosing strategy, which could be done, or we could start to do some testing in humans as well. I think that's a really good question for any intervention right now. I mean, the other thing is with the AKG, we really only tested one dose in the mice, and so, you know, I think that higher doses may have even more positive effects. With AKG, it's been tested up to 10 grams a day in another clinical study for osteoporosis, and they found no evidence of toxicity. So as far as we know, there's nothing terribly risky about taking more. So we want to go back and do mouse studies now where we ramp the dose up. The other thing I'll say, dosing is another thing that's very personalized. And if you look at the vitamin studies like vitamin E and vitamin A, everybody says we did a meta-analysis and we don't see any benefit. Well, go back and look at those studies and ask how many times they actually measure the levels of vitamin E and vitamin A in the individuals in the study. They just gave us a supplement to people. They took it and they measured effects. They generally didn't try to optimize the levels of the vitamins. And so our studies on vitamin A is an interesting molecule. It's beneficial. It does a lot of good things, but people call it a vitamin, so it's safe. If you take too much, it's toxic. People have known that for a long time. And so we've been playing around with the dose in mice, and we can get as much of a 20% lifespan extension with just vitamin A, but we got to get the dose right. And so what we're doing now is going back and doing personalized dosing. So each mouse gets a different dose of vitamin A based on efficacy and toxicity markers in the blood, and we keep modifying that dose. And my guess is if we can optimize, even just with a set of vitamins, if we can optimize levels, we can have a profound effect on aging in an individual. Of course, that gets more expensive, right? Because you have to monitor the levels of things. But nonetheless, I think there's a lot of room for improvement with just things that, vitamins and things like AKG and maybe NAD precursors that, we haven't really done any of those kind of dosing tests or personalized dosing. And so there's a lot of room for progress there.
No, exactly. And personalized medicine is something that's also I find really interesting, because you're right, we are all different. We have different genetic variations. We all have different environmental factors that we're exposed to. We do different jobs. We eat different things. And so I guess another question is, if we're going to kind of strive for like personalized medicine, what kind of things would we need to do in terms of biomarkers? I suppose the better way of phrasing it is, what would be like the gold standard for biomarkers to be able to achieve personalized medicine?
Yeah, I think that's an interesting question. I mean, you could imagine optimizing the aging biomarkers. But one thing about aging biomarkers is that we tend to think that biologic aging behaves like chronologic aging. Chronologic aging is just measured by time and goes steadily up. My guess with these biologic aging markers is that's not what's happening, that they're oscillating. And that depending on how much sleep you got, whether you ate healthy, did you go for a run, you have the virus, and you might be biologically 10 years different from one 2-week period to the next. And so I sort of see it as, as you get older, that oscillation may be increasing, and eventually you cross some threshold, and then you're also hyperglycemic or something, you get diabetes. So I think that we really need to look longitudinally at these biomarkers, because one time point may not give you a really good determination of what your biologic age is. So if you're optimizing to that, it might work, but the oscillation may cause problems. So what we're trying to do right now is, for things like vitamin A, we know what the toxicity markers are, so we look at those. But we don't really know what vitamin A does to aging, so we're trying to identify the right efficacy marker too. So we're looking at inflammatory markers and other things. But I think that's one challenge right now to personalized drug dosing, is that we don't always know what we want to optimize for in the blood. And so there's a little bit of science that has to go in to figure that out too.
So what are the kind of main biomarkers that you think at the moment are most effective? So I know there's a lot of talk about using the epigenetic aging clock. So would you be able to explain a bit what that is?
Yeah, so even around 2010, everyone was saying that the search for aging biomarkers is basically a failure. We have things like walking speed and grip strength and time get up and go and a few other things that work particularly well in elderly populations. But in terms of molecular biomarkers, none of them were that predictive. Even telomere length, I mean, it does have some correlation with life expectancy, but it's not that strong. What happened is that people like Steve Horvath and others, they started collecting really deep data sets of genome-wide DNA methylation, and then had the person's age. And so they put age into the computer and then had the computer use machine learning to optimize the state of methylation at all these different sites in the genome to predict a person's age. So if your age is 50 and the biologic age says you're 45, that really what that is the error in the algorithm, because the algorithm was trying to predict chronologic age. But the argument is that the error is there because it's not really, can't really measure chronologic age, it's measuring biologic age. And so what that's saying is that you're actually aging very well. You're only 45 when you actually have lived 50 years. And that now there's a whole range of different evolved methylation clocks. And that's one of the things we're using in Singapore. I'll say, however, that it's still unclear whether there's anything special about DNA methylation. There are clocks based on transcriptomics, there are clocks based on microbiome now, there are clocks based on metabolomics. And so it may be that, and like I said, there's a facial pattern clock. It may be that anything that you can get enough complex data and enough people and use the right AI, you can predict biologic age from. And so I think that's still an open question. Is there anything special about methylation? But we're using the facial pattern stuff. We're doing complete blood counts because there's a company called Gero that's can do biologic age from that data. We're collecting accelerometry data. We're collecting DEXA scans and immune markers and some of the more classic clinical markers. And the idea is to really merge all that together and see, what we're getting out of it, and are we looking at, several sides of the same coin, or are we looking at completely different things, which would be kind of scary. So I hope that's not the case. So it's the first step is really to bring these different biomarkers together and see how they interact.
That's a really good answer. So I agree. I think the improvements as well, that it seems like every few months there's a more like improvement upon these different clocks in terms of their accuracy and their ability to integrate the different markers together. And so I guess one issue in terms of being able to make these biomarkers scalable is to have a kind of cost-effective biomarker. And so also the facial patterns is something that anyone could just take a photo. It seems quite easily scalable. But in terms of like these more omics approaches, how scalable do you think they are?
Well, first of all, the collaborator we work with on the facial pattern is Jackie Han at Peking University. And it's actually was originally optimized in the Han Chinese ethnicity. It doesn't work with just a phone picture yet. You need to do a three-dimensional reconstruction that your phone can't quite, it's getting close, but can't quite do. So it's not as simple, but I think phones will get there, and the technology may improve to use phone data as well. There are other people that have tried to generate clocks from phone pictures as well. But the other stuff, methylation can now be done from saliva. So with the that's why we chose the particular methylation clock we did, is you could send an envelope with a piece of paper along with AKG. This is for the company, and you could spit on the piece of paper and send it back in, and the DNA is stable, and so that's very scalable. If you have to take a blood draw, it gets a little bit more complicated, and we've just completely rejected anything that requires biopsies, because while they may be more informative, I can certainly do a lot with biopsies. I can't, people aren't going to give muscle biopsies. You just won't do it. So, and you can't scale it anyway. So I think that we're willing to look at those more complex things or things like DEXA scanning, but that's really more for research purposes. The main goal of the biomarkers is, again, something that everybody can do.
Cool. So, in terms of future research, are there any particular areas that you think are very promising in terms of discovering new potential interventions or improving our understanding of aging? For example, like regenerative medicine approaches. Is there anything you've kind of got your eye on that you think looks very promising?
Yeah. I think that there's a lot of futuristic things, gene therapy, regenerative medicine, and we don't have any idea yet what can be accomplished there. Reprogramming, there are studies now saying that you can reprogram adult cells and that has an effect on aging. There's certainly some potential risk there, but I think that could, in theory, that could cause a dramatic change to life expectancy. So I think that we'll continue to get new and better and technologically advanced things. One of the things that I want to study right now are mesenchymal stem cells, because people go get these treatments all over the world, pay a lot of money, and a lot of people will tell you they have a tremendous effect on their aging and their inflammation and things like that. So, but there really aren't any good biomarker studies on aging. So we're looking to partner with a company to try to achieve that now. We may not be able to do a full placebo-controlled study, but at least we can baseline it and look at the follow-up, because you can easily imagine how something like mesenchymal stem cells and the factors that these cells secrete that are anti-inflammatory and stimulate your own adult stem cells might be beneficial. And so I think that right now I'm still trying to corral all the potential interventions out there and see what works. But I think there are a lot of exciting things that may have. Rapamycin is probably the gold standard right now, but it may be trivial compared to what can be done in 10 or 15 years. The field's really taking off, and there's a lot of private sector money in it now, which is great. So it allows crazy companies doing different things, and it's a little bit the Wild West, but some of these things are going to work. So I'm excited to see where the field goes.
Good answer. So could you actually explain a bit more, for those who don't know, what mesenchymal stem cells actually are? What are they proposed to do?
Yeah, so these are stem cells that are isolated from bone marrow or other places in your body. They respond to damage or injury honed to those sites, and they have an anti-inflammatory. They secrete factors that are anti-inflammatory. They may, to some extent, in your body contribute to repair of the tissue, but it looks like that they don't have such a big role in that. It's more stimulating the local cells to do the repair. They are somewhat immune protected, so when you get an injection of them, you don't have to have your own cells. And so a lot of these companies now are taking like cells from umbilical cord, isolating the MSCs in the umbilical cord, growing them out and injecting them into people. I think that there was a lot of hope initially that when you put these cells in, there was a lot of studies like in heart failure, and they were hoping that they would engraft into the heart and replace the damaged tissue, and that didn't really happen. But what they found was that these cells actually had a beneficial effect even though they weren't engrafting. So that's when people started looking at these factors that are secreted. And so they hang around in the body for a period of time. It depends on who you talk to, maybe up to 6 months. And they may be providing some really beneficial effects. But like I said, a lot of athletes take them for soft tissue repair. They stimulate muscle repair, but we don't have, I don't think, really good aging data yet. And so it would be interesting to find out if it's really affecting biologic age.
Cool. So we've spoken a lot about a lot of exciting areas of current research in terms of different interventions. But in addition to the exciting work, are there any major hurdles that the field kind of still faces in general, like funding being one of them?
Well, government funding is still lagging behind. There's still a lack of awareness that aging is something we need to deal with, and it's probably the most important thing to deal with. I mean, most of the medical world is focused around sick care. We wait till you get some chronic disease and we try to treat it, and that costs a lot. It's not that effective, but people make a lot of money off of it. And so there's an entrenched system to treating people, and converting more to a preventative medicine in healthcare is a real challenge. You know, the system is not set up to deal with that. It's hard to do clinical studies. The pharmaceutical companies, you know, if we kept everybody healthy, all their drugs for sick people wouldn't make any money anymore. So the field is full of people that want to do good, but they're also driven by money. And so, you know, it's a challenge to really convert back to a healthcare system. You know, I tell people when I get sick, I want to be treated, but I'd rather just not get sick. And I think it's going to be easier to slow aging than it is to treat most of these diseases of aging. But we have to commit to it, and there's just not enough government funding right now. The other challenge is that aging is still not recognized as a disease. You know, it's really crazy, right? When you think about it, cancer affects a third of the population, and we spend trillions of dollars trying to do something about it. Aging affects 100% of the population. We're like, oh, it's natural. Yeah, it's like, you know, if buses ran over 30% of the population and trucks hit 100%, we'd regulate the buses. You know, it doesn't make any sense. And so, but that's, you know, I think people spent thousands of years rationalizing aging. And oh, it's good, it gives you wisdom, and there's religious arguments for it, and it's natural. And I think we need to let that go. I mean, I think we can get wisdom as experience, right? If we can slow aging, we can have people with a lot of wisdom that are still young. And so I think we need to just change that mindset, make aging a recognized target for intervention. And also just take a full life course approach to healthcare. You know, I think that if, you know, I walk by the Hippocratic Oath, there's a big statue of it in US medical school. And this is a great document, okay, but the way it's interpreted now is maybe not quite right. You know, the first thing people say when you ask what that means is first do no harm. And the way they interpret that is that if someone's healthy, we shouldn't really take any risks with them because, you know, we can only do harm. But I don't think anybody's completely healthy. You know, if you have somebody that's 40 and doesn't meet the criteria for a clinical diagnosis of disease, there's still a lot you can do to help those people. And by doing nothing, you're probably harming them. So I think that we need to take more of a life course approach to medicine. We need to encourage healthy lifestyles very early in life. Maybe look at supplements as you approach middle age. And maybe even more radical things as you get closer to getting chronic diseases, or even in people that already have chronic diseases potentially. And if we do that, we'll have a much bigger impact on health span in the long run.
That's a really nice way of describing it. So where would you hope healthcare to be in, let's say, 10 years' time?
I would love to see widespread adoption of strategies to extend health span. And, you know, you can argue that already sort of exists. You know, like the Health Promotion Board here has an app that they give out, and wearables that they give out to try to encourage people to take more steps. So from a lifestyle perspective, people are already trying things. The problem is that getting people to do sustainable lifestyle change is not easy. But people are trying those things. But I'd like to see supplements and even drugs added to that list. So we can really try to have, I think it's totally feasible to affect health span by 5 to 10 years. But it has to be something that someone's willing to commit to and really make the effort. And ultimately, it has to be a combination of private and public sector approaches to solve this problem. And so there's still that battle of changing the mindset of saying that, you know, just because everybody ages doesn't make it something you want to do. And you know, and so I think the whole field is still struggling to get that message across.
So speaking of interventions, is there any advice that you'd give to people now if they wanted to help improve like their health span or their health in general? What would you advise?
Yeah, I always sound like your mother when I answer this. But also, yeah, there are really 4 things that you can do right now. I mean, if you want to try supplements, great, try them. I mean, we're still finalizing some validation on a lot of things, but there's a lot of potential candidates. But outside of that, it's about lifestyle, and there's really 4 factors. There's exercise, some combination of cardiovascular and resistance exercise with resistance training, healthy diet, which we could talk all day about, but don't eat too many calories. That's a good start. Maybe consider some of these intermittent fasting strategies. And then I would argue to have a balanced portions of macronutrients in your diet. Stress. It's kind of hard to study this in animals, but I think the amount of stress you feel is a big driver of aging. And then sleep, which is not that easy to modify sometimes, but sleep quality declines with age, and I think it's important to try to mitigate that. But the question is, how do you do that? And ultimately, it has to be changes you can make that are sustainable, which means that you enjoy them. If I tell you to be a vegan marathon runner, you're probably not going to do it. So I think it really takes people to think about it's personalized. People should think about their lives, look at where they can start to make slow changes that are toward health. And I think if they do that, it'll keep growing, because what you'll realize is you feel better now, not just 20 years from now. And that's especially true of exercise. There's a threshold you have to get over before you really feel the benefits of it. If you don't exercise and you start running or doing something, the first 2 months is mostly misery. But once you get over a certain threshold, then you find that it feels good to run, and it actually takes away stress to run, and it gives you more energy. And so, but you have to get over those barriers. The same thing's true of like taking sugar out of your diet. If you talk to people that switch from Coke to Diet Coke, they hate the Diet Coke at the beginning, and 8 weeks later they hate the Coke because it's way too sweet, but it takes time. Your body doesn't transition quickly. So it's about sustainability, and that's the key for lifestyle.
That's really great advice. So thank you very much for taking the time to speak with me today. It's been great to kind of pick your brain and to find out a lot more about these clinical trials that you're conducting in Singapore. So unless you have any final words, it's just been fantastic to have you on the show today.
No, thanks for having me, and it's great you're doing this show. I think it's wonderful.
Thank you very much. So I really hope you've enjoyed listening to this interview and have learned something today. I want to say thank you to my Patreon supporters, and thank you for listening.